All right, so we've talked about some vascular tissues in the vascular land plants, and now we're going to start to talk about seed plants. And the first group of seed plants we're going to look at here and talk about are the gymnosperms. So the gymnosperms actually represent several phyla, and the particular phyla that's the example we're going to look at here are the conifers. But for all the gymnosperms, this kind of means naked seed. And that's kind of where we're going to start. We're going to talk about the gymnosperm seed itself and some of the structures there. And then we're going to go through the process overall and naming some of the structures, cells, and tissues along that process. Before we really get into that, we'll talk about what's the purpose of a seed, and that is it allows the organism to then a couple things. One, move further into land, so there's not as much worry about desiccation. So what is the seed, really, or what do we have inside the seed? Well, we have, and this is representing a gymnosperm seed here. One, we have the plant embryo. So a multicellular 2N embryo that's developed from fertilization of a male haploid nucleus and a female haploid nucleus fused together to form a zygote and then develops into the embryo. We then have, surrounding it, gametophyte tissue. All right, and so the organism, the embryo, will grow into the sporophyte organism. So we still have the alternation generations, gametophyte, sporophyte, and in this case here, the sporophyte is kind of developing inside what is the gametophyte tissue, kind of nourishing it. So we have that. Then we have surrounding that, there's going to be a seed coat here. That will help protect it. This whole thing here is the seed coat, right? And then inside the seed coat here is a spore cell wall that provides additional protection during development. So this can then stay dormant. So this is another aspect. Not only can it be dispersed without drying out, but it can stay dormant for a very long period of time, years, before it may then continue its development and emerge into a seedling and then develop into a more larger adult plant. So the seeds can stay, the organisms can stay in this form for a very long period of time. So it's for both dispersal, so distance, but also preservation in time, right? So that they don't have to immediately develop because they have nutrients there and they have ways of altering their metabolism until the environmental conditions are proper, so they can stay in this form for a very long time until conditions are good. As we get into some of the discussion of tissues and a lot of the terminology, one thing I've found is that it can become very confusing overall. And this is not a botany course where we're going into all the details of everything. We're trying to cover everything from genetics and population genetics and mechanisms of evolution, fossil record and evolutionary history, to prokaryotic organisms and differences between the different domains of life there, the eukaryotic organisms, plants and animals and fungi and ecology. And so we can't really get into all the details. It's not realistic to even ask that is a student be responsible for it as well in this particular course. So I'm going to give you the overview. There's a bunch of structures that I'm going to talk about. Some of them a little bit more than what you're going to be responsible for. And some of the things I'm not even really going to talk about or mention because there actually is a lot more detail that I'm even just going to completely skip over. And at the end, I'll kind of go back and emphasize what is it that you actually would be responsible for in an organismal biology class like this, where we're covering everything and not just plants. So this is the particular focus of this lecture right now, is on the conifers. So these are going to be our cone-producing plants. Okay, so this is called an ovulate cone. This is an ovulate cone. Okay. And so they range in size, and some could be much smaller than this one. And this one is very large. It's one of the larger ones that we have. And these are going to be these little scales, right, that we're going to talk about here. It's going to be actually this structure here is kind of where we're talking about these sort of scales. This is where the seeds are going to be held. So this is a female ovulate cone. The male cones are very small. I don't actually have an example, and you probably wouldn't be able to see it. And they do break down a lot quicker, so they're more difficult to find in a preserved state. In relation to that, talking about the male versus the female cones, there's going to be a lot of terminology that is brought up that I think if you just kind of have the basics of the terminology, it'll really help. So when we talk about terms like anther and arche, these terms like antheridium and archegonium and so forth, we're talking about tissues in plants in general. And anther tissues are usually referring to male tissues, whereas the archae tissues are referring to female tissues. So antheridium tissues would be then making sperm, and archegonium tissues would be ones producing an egg, generally. We're also going to have terms like mega and micro, and usually that also implies like large and small, but in this case as well, mega is typically going to also be referring to the female, whereas micro will be referring, not that male, referring to male. So like microspore or microsporocyte or microsporangium, all those terms are related to male structures, whereas megasporocyte, megasporangium, megaspore itself, those mega all terms are referring to female structures. So keep that in mind. And then as we break down some of those terms, again, spore versus sporocyte versus sporophyte, like sporophyte plant. Okay, so phyte, referring to plant. Cyte, we're referring to a cell, right? So a sporophyte is the spore-producing plant. The sporocyte is the spore-producing cell, right? So cells that are going to then divide to produce the spores themselves, right? The spores are going to be then haploid structures, generally. Gametes are also going to be haploid structures. The organisms then that are the gametophytes are the organisms where all the cells in those organisms are haploid. And the sporophyte are going to be the organism that, while they produce the one-end spores, they're the ones that actually can undergo meiosis. So this is going to be through meiosis, because the sporangium structure itself that'll undergo the process to produce the spores, and the organism itself, the sporophyte, is a 2N organism, so a diploid organism. So there's so many different terms. That's really the thing that you will be responsible for as a first step, is really just terms and definitions of terms and keeping a lot of the terms straight, right? And then we're going to apply some of those terms to process, and not every aspect of it, but the overall big picture, and being able to identify and label some of the key or most important structures that are involved in that process. And that's what we're going to kind of get into now. So as we come over here, we're going to look at the life cycle of a conifer in general with some simplification, because even in some of these things I pre-sketched, I haven't included everything in it. Some of these things there'd be more of them. There'd be 2, say, of each of these little ovules. And then sometimes there's more cells being produced. There's 4 cells, and I'm not drawing all of them because it just becomes a little too much. So don't worry about all those details, and don't expect that it's going to be completely detailed as it would be in an actual botany course. This is more just for the comparative aspect of our organismal course. So here we have an adult tree, and this is some adult conifer. Wow, is it not working? A conifer, and this would be the sporophyte. So let me quickly try and find the other marker because my marker is dead. So 2N, sporophyte, plant, large, dominant. So part of our focus is on differences and comparative differences between different groups of, say, plants, right? So we have non-vascular plants, then that are they mosses, right? Then we have vascular plants that begin with the group, with the groups that include the Pterophyta. There are a number of other phyla there as well, and then we start to have the vascular tissue. So the gymnosperms are vascular plants as well, and then the angiosperms that we get into, they're vascular plants as well. The gymnosperms and angiosperms will be, in addition, seed-producing plants. So they're vascular and seed-producing, whereas the ferns are vascular but non-seed-producing. So that's why we're kind of emphasizing the seed now and what the seed structure is. So it's kind of those comparisons. They have some similar aspects to the life cycles, but then some things that are different or unique. So sporophyte plant here is the dominant one, as opposed to the gametophyte plant, which was the dominant one for the mosses, for example. So you kind of have that kind of comparison. On these plants, there'll be some of the branches where we have the needles, which are their leaves. And then eventually when the plant reaches maturity, they'll start to produce the cone structures, right? So what we have here is the female or ovulate cone. And this is representing a male cone. Now, size-wise, I drew this one a little bit bigger, but in reality, some of them would be, this one would be very, very, very large, and the male one would be really, really, really small in this particular case. All right, and this is the pollen cone, the male one. So, looking at the male cone, all right, there are then going to be structures in the male cone that are produced called the microsporangium. All right, these structures that are then going to produce the pollen grains, and so this is where meiosis will take place, and will produce pollen. So we're going to put this here. I'll put it here in the middle. All right, so the pollen grain. Inside the pollen grain are really going to be 2 main cells that we're going to follow more of their fate. All right, and the one is really what will become the sperm, and the other will become the pollen tube. Which we'll kind of get into here in a second. So the pollen is the dispersal stage for the male. The female doesn't really have a dispersal stage. So the unfertilized egg, the ova, stays inside the cone. Whereas with the male, the pollen is distributed from the cone. So that's the first aspect of this. There's a cone for the male, there's a cone for the female, but the haploid pollen here gets distributed and is carried by the air great distances. So this is one of the things that allows it to be an advantage of a land plant. Great distances of travel. Often I kind of drew this with a little wing-like structure on it because many of them actually do have structures that allow them to be picked up by wind and carried so they can travel really far distances. what has to happen is they obviously have to fertilize the ova, so they have to get to female plants. Now, in the ferns and mosses, our previous groups of seedless plants, they had a sperm with a flagella that swims in water, so they were completely tied to water. Right? So they cannot reproduce unless there's a lot of water around, so that you're only going to find them in very moist environments. So you wouldn't find them in a desert, for example. Because there just isn't going to be enough water there, one, to distribute them and to allow the fertilization process to even take place. Here, it's not relying on water, so they can disperse much further from water sources and get into much drier environments. Obviously, they're going to need water at some point, but they don't need it for fertilization is the point. So the pollen is distributed by air, and eventually it'll reach then a female cone, right, the ovulate cone. And then the pollen will come into something here. So here's kind of like a zoomed in example of it. So this is from the female cone. This is the ovule. That's not so visible. Here, and this is representing the pollen. And they don't. There's an integument here, covering on the outside, but there's an opening right here called a micropyle, P-Y-L-E, micropyle. All right, and that opening is then what allows the pollen to enter this area to begin the fertilization process. What will happen is once the pollen enters, the cells that are destined to become the pollen tube will start to undergo mitosis, right? Until they have more and more and more cells, and they will start to then develop this structure that I have drawn in here, all right, called the pollen tube. Now that can take a year to happen. So it's not a fast process. It's actually a very slow process. Once that tube then connects here with the coat that's around the unfertilized ova, then the nuclei from sperm will then travel here and enter, and then fertilization can take place to then produce the zygote, which will develop into the embryo through fertilization, or 2M. So, but that could then that'll take potentially another year or more. So the overall process from the pollen reaching here, the ovulate cone, and then undergoing fertilization, and then the continued development of the embryo to actually produce the seed, is an overall almost at least 1 to over 2 year or more process, depending on different organisms, different species. It's hard to always put out rules because you'll always find something that does it slightly different. But it's always going to be a very long process, lengthy process when it occurs. So the pollen is distributed by air. It gets to the female ovulate cone. It can then build a pollen tube. The pollen tube then connects with this structure that is then going to be producing the egg, or the ova, the unfertilized egg, and then eventually the sperm nucleus will come into it, and then the fertilization process will occur. So that's kind of the male part, kind of in a simplification with a few terms. For the female part, so kind of zoom in a little bit here, the ovulate cone. What we're going to have here are a number of structures that are going to be produced per little scale. So these are these scales. So that's kind of like these. That's what we're talking about here. These things here are the scales. And so inside, each of these, we're down there at the base is what we're looking at here, is where the megasporangium is, and then the sporocytes will develop, and then the ova will develop, and all these things will actually happen in here. The fertilization process will happen in here. Something else to kind of note that's very interesting is when the fertilization process starts, these scales will actually close and they'll actually be shut. And then over time, once the embryo then starts to develop and the seed is ready to be distributed, produced, then the scales open back up again so that the seeds can come out and be distributed. Some conifers like this actually require fire to activate and distribute the seed. So if actually fire doesn't occur in a particular area, the seeds won't be dispersed and they can't even then develop. So different plants rely on a variety of different environmental processes that if humans interfere with can actually disrupt their life cycle. Even if they seem to be bad, like fire, that's partly a totally separate topic, but over managing a forest by not allowing natural fires then creates really bad fires that then might burn too hot that destroy the entire forest, as opposed to letting smaller ones that are necessary for certain plants in their reproductive process. In any case, kind of moving on, so we have this tissue here, this megasporocyte, okay, which is the cell itself, and then we have the megasporangium, which is kind of the tissue then surrounding it. So if we think back about there's the marker that I was looking for the whole time that I kind of lost. All right. So the megasporocyte, things to add in here. So this is again a 2N, structure, and so is the sporangium. All this stuff is a 2N or a diploid structure, but that's again where meiosis will take place eventually, and then this is a process where it can be a little confusing, and there's a lot of extra things that happen. Often meiosis doesn't take place to actually produce the haploid ova until after the fertilization process begins. So until the pollen attaches, builds the pollen tube, and the pollen tube connects, then that would actually be triggering the meiosis process so that you can get the fusion. In addition to that, there's a fusion of the 1N nuclei, another 1N nuclei from the pollen, with 2N cells to actually produce a 3N coating that we'll talk about a little bit more as we talk about the angiosperms and seed plants, but we're not going to talk about that right now. So here we have an archegonium, which is a larger structure. And remember, arche means the female and the egg producing, right? So that's going to be what produces the egg. Here's where we have the pollen and the pollen tube. Pollen and the pollen tube, here. Typically, when meiosis takes place, there'll be one cell that's produced there's 4 cells that are produced through meiosis. We've covered that before. One of them remain and then continues to develop into the ova, whereas the other 3 end up being broken down and disintegrating. And that happens in some cases with the pollen, with the sperm, as well. But not all of them are going on from meiosis. It's not that meiosis only produces a single cell. It produces others, but they don't all have the same fate. Only one typically does. So this structure will start to develop, and then what we'll get is the female gametophyte. And the significance of the female gametophyte tissue is that it's going to be, first off, multicellular, and haploid tissue. And again, that's the tissue here that's eventually going to remain and surround the embryo to provide nutrients, right, for the embryo itself. And so at this point in time, this is where it's forming from these haploid cells after meiosis. So some of these cells end up becoming the ova, all right, the unfertilized egg, and then some of the cells end up becoming the gametophyte, which then they will undergo mitosis and continue to develop to become multicellular, whereas obviously the ova, after it's produced, just stays as a single cell waiting for fertilization. And so then we have fertilization actually occurring, where the pollen comes in, produces the pollen tube, and then eventually the nucleus is passed on to the ova, and then it can start to develop. And so then the embryo, so the single cell. So there's another term here that I've been saying, and you may not be familiar with it. So it's zygote. The zygote is a 2N cell. All right, it's the result of fertilization. So you have 2 cells, the female, 1N egg, the male, 1N sperm. They then join together. Typically, the sperm doesn't enter the egg. It's just the nuclei that enters it. Then those nuclei join together, and that forms a new cell that's 2N. So it's pretty much the same cellular membrane, cellular material that's all female. The male material doesn't really come in. Typically nothing else comes in except the nucleus. And then that cell itself, that new is called a zygote. As the zygote cell starts to then divide through mitosis, it begins forming the embryo. So the embryo is multicellular, 2N structure that is forming from the zygote, from the fertilized ova. And in this particular case, we also refer to it as the sporophyte plant, because it's the 2N plant, and it's going to be the sporophyte generation that will eventually produce the spore structures and cells and so forth. Typically in these plants as well, what will happen is in the scale, you'll usually have the ova continue to develop and the seed itself. So now we're looking at this structure kind of simplified, like a lot smaller. There'll be, for dispersal, some kind of modification to the scale, or a scale that's kind of attached to the seed, that then almost like a little wing here. That's what these should look like, hopefully, to you. And the idea is that when the plant has the cones open back up again, and wind or something else is used to disperse the seed, that it can fly a lot further, or be moved a lot further away from the parent plant to reduce competition, and to then start to spread the species into more and different habitats as well. So the ability to have a greater dispersal distance, the potential for greater success, because they'll be trying out different habitats, and then they won't be competing with each other. So you have a sort of a scale modification that's like that lift it in the air to provide to be carried by the wind, essentially. So this is a type of scale. And then what will happen is eventually the seed is distributed, will find a site to settle down, and then it may develop or it may not if the conditions aren't right. So it can stay dormant for a very long time. So remember that is one of the significant adaptations. Dormant for how long? It really just depends on the conditions. All right. But eventually then the embryo will burst out of the seed coat, and then the new little sapling, that's what it's called, which is also a whole multicellular organism. It's 2N. It is, again, the sporophyte plant that's now dividing and growing at just an early stage. And now you start to get the development of different types of tissue. So you get the root tissue, the shoots, and then the leaves, and all the vascular tissues will start forming, and all these things, which some of those we've already talked about, and some we'll continue to talk about a little bit more. And then eventually it grows up into the adult plant. Once it reaches maturity, can then start to produce the cones, which will then undergo this process to produce micro or megaspores, and then continue on. So, I said at the end I would kind of give you the overview, like what do you need to know now, because it starts to become a lot of material. And as I said, if you're looking at this for a botany class, there's a lot I'm not including in here. There's way more information. There's other things that happen. There's a lot of other details and terms that are not being covered. So you'd have to look elsewhere for that. But for just this general organismal class, talking about What are gymnosperms? They're seed-producing plants. Our example of the gymnosperms, we're looking at the conifers as the phyla, so that's the actual phyla that we're looking at, Conifer phyta. And then we're looking at starting off with the seed. So we got the seed. Inside every seed is an embryo. So in our particular class, we did some germination experiments. Took a seed from a pea, or essentially a little pea. The pea is a seed. Inside each pea is a little baby plant, right? And inside that, or surrounding it, is then nutrients. So that it has a protective coat, and then it has its own little nutrient store, right? So the nutrient store here is from the gametophyte tissue. So it kind of lives within the gametophyte tissue until it's ready. Often needs water to stimulate the germination process. So it'll usually, water will come in, the structure will swell, usually the seed coat will then open up, and then the embryo can move out, the tissues will then start to grow, and then you'll get the root system and then the shoots, which hopefully you've seen firsthand with your little pea plants that actually germinated from those seeds. They're not conifers, but still the same thing happens because they're seed plants. They're all seed plants. So that's the seed structure. One thing else, a term that I would like you to know is the micropyle. So that's kind of in the unfertilized ovule, that's kind of like an opening here that will allow the pollen to move in. So kind of then getting back here, what should you know? There are two types of cones in the conifers, right? There's the female and the male. The female is called the ovulate cone. The male is called the pollen cone, right? The pollen cone then has two end structures called micro. Remember, we said the micro is typically the male, sperm producing, or in this particular case for dispersal, we do not have a swimming sperm. We have pollen that's carried by the air. Right? And so inside the pollen are typically a few cells. Some of those cells will become like the sperm that will do the fertilization process where the nuclei will be transferred, but other cells in there will create other structures necessary for fertilization. And the key structure there is called a pollen tube. So the pollen actually produces its own tube, and we said that's a fairly long process, taking often a year in time from the time the pollen grain reaches the ovulate cone to build that tube to then begin the fertilization process. So it's very slow. Inside the ovulate cone, the female cone, you have a megasporangium. Okay, so that's to be egg. Mega would be then the female. It's going to be then the egg producing ultimately. Archegonium, arche, is going to be the actual structure that produces the egg itself. Again, but in this case, egg, I also keep referring to it as ova, because ova is really the unfertilized egg, the haploid. And so then what happens here is there's actually many more things that go on here. This is the part that I said I was really simplifying or skipping over, because you'll have multiple ova being produced in each archegonium, and there's a lot of other things, but don't worry about that. Just kind of the basic terms and the general process. Okay, so that will be produced. Some of the cells there after meiosis occurs will then lead to the ova. Some of the cells will then form the gametophyte plant, which is in this particular case just stays there around the embryo and provides nutrients for it. Right. So that's what's going to happen there. Eventually a seed coat, is formed that is going to protect it from, and that's derived usually from the integument of the plant itself. And then there's sort of a spore wall coat that's produced by the this structure here, that's like the sporangium tissue that then ends up forming that coat. So you have a couple coatings that protect it against ultraviolet light and desiccation and other environmental chemicals. So there's a lot of protection for the individual seed. And then after the development process occurs, which can then take another year or 2 years, the seed is ready to be dispersed, which again also happens typically by the wind. And so it has modifications in structure, so it can easily be carried by the wind with these sort of wing-like scales. That should cover it with the main terminology, some of the breakdowns of some of the terms, and the main things that you need as the takeaway from this. And then you'll have term lists to kind of go through as a checkoff to kind of see that you have all those terms, and you can write a definition for each of them, and then being able to label some parts of this, is which is what you may see then on your exam. Some drawings like this that are just unlabeled that then you have to kind of fill some of it in.